Hello everybody, I'm going to go over test 4 today. Another routine, test 4 from fall 2013. The first 30 questions on this test are from Unit 10. And these are all the slide references on the left-hand column. Final 10 questions are from Unit 11. And start going through these here. So which enzyme is essential for the digestion of dietary lipids and the synthesis of eicosanoids? So that's going to be phospholipase A2, or PLA2 is the other way. You might have it written in that. So digestion of dietary lipids you can find on slide 9, and the other two are talking about eicosanoids. So which molecule facilitates the digestion of dietary lipids? It's kind of on one random slide by itself. It's the bile salt, glycocholate. Everything else doesn't really have anything to do with dietary lipids. So it's just a matter of can you recognize what the bile salt is. Lipoprotein particles are responsible for reverse cholesterol transport. That sounds like a good thing, so that's the good cholesterol. Let's see, on this slide you'll find out that IDL is the LDL precursor, chylomicrons transport fat, LDLs transport cholesterol, and VLDLs do endogenous fat. So make sure to know that in that column. Which enzyme starts the mobilization of triacylglycerols? That's going to be ATGL. This one's easy. Targets the monoacylglycerols. Hormone-sensitive lipase targets the diacylglycerols. This is the activator of fatty acids, and AMP kinase is mentioned briefly in the end towards the regulation slides. Not much to do with the mobilization. This molecule is not needed for the reassembly of dietary lipids. So if you go to slide 10, you see that you take a fatty acid, use an ATP to put a CoA on it, and cholesterol is also used in the reassembly. Water is not used. Water is used in the mobilization of dietary lipids, but not the reassembly. Which reaction is part of the reassembly of dietary lipids? That's also in slide 10. It's the activation. Fatty acid activation. Everything else, carnitine involved in the shuttle. Which protein has not yet been found in red blood cells? So remember, red blood cells have no mitochondria, and the carnitine shuttle is a mitochondria cytosol shuttle. Or this carrier is on that mitochondrial membrane, so it would not be found in red blood cells. Everything else can be found in the cytosol, so therefore it can be found in the red blood cells. Which enzymatic process does not take place during the beta oxidation of palmitate? He's always going to ask about palmitate. It's his favorite. 16 carbons with no unsaturations. So yeah, that would be a saturated fat. So you definitely need the activation from the synthetase. You go through oxidation, thiolysis, also the hydration. You don't go through a hydrolysis. The transfer part is talked about on slide 34. There's a couple different things where he could be talking about the actual transfer. There's technically 2 transfers involved. But again, hydrolysis is for the mobilization. How many molecules of water are used in the beta oxidation of palmityl-CoA? So there's 1 hydration per round. So if you're doing number of rounds, 16 minus 2 divided by 2, that's the number of carbons, minus 2 divided by 2 equals 7, so 7 is your answer. Which is not an intracellular enzyme? So there's two ways to know this. You can know you've seen all of these before and they are all in the cells. And lipoproteins are what's being reassembled into your lymph system, then transported into your blood. So I looked through, I couldn't find lipoprotein lipase anywhere. That might be a process of elimination. Those lipoproteins are blood transporters, or transporters in the blood for the triacylglycerides and the cholesterol and everything. So that's going to be the answer there. Let's see, on slide 9, there's some digestive lipases. And those would be found in the lumen of the digestive system, so those could be an answer as well. There are some lipases in the fat cells. Fat cell lipases that are involved in mobilization, lipases, and those are found on slide 34. Make sure you know where all the enzymes are in this unit. It's a popular question. It's come up a couple times already. Which vitamin is not needed for the beta oxidation of palmitate? Going to be biotin. Vitamin B3, 2, and 5 are going to be used for all beta oxidation. Biotin is only used for that carboxylase that's used in the odd chain fatty acids. Along here, that carboxylase also uses an ATP. That's another thing that comes up a lot. So odd chain fatty acids require that carboxylase, and the epimerase and the mutase. I'm pretty sure it's on one of these two slides here. Which metabolite is not synthesized in the brain? So now we're getting into some ketone body questions. Ketone bodies, acetoacetate being the other main one, are going to be found or synthesized in the liver, not synthesized in the brain. The brain will use this for energy, but it won't synthesize it itself. Which metabolite is particularly elevated in the blood during starvation? Lactate shouldn't change that much. Glucose is going to go down. Oleate, or any fatty acid, is going to stay the same, slightly go up. But either of the ketone bodies, hydroxybutyrate being the other one, are going to go way up. So that's the answer there. Which of these molecules does not have a mitochondrial carrier? That's what kind of threw me off, because I initially was like, oh, fatty acyl CoA has the carnitine shuttle, but we're talking about carriers / transporters versus shuttles. So fatty acyl CoA does not have its own carrier, does not have its own transporter. It uses the carnitine shuttle to get in. If you go back to Unit 6, it lists the mitochondrial carriers here, and all of these have their own carriers. Citrate's a tricky one because there is the citrate shuttle that we talk about later in the unit, but that citrate shuttle has nothing to do with using the citrate. It's much more about getting acetyl-CoA from the mitochondria out into the cytosol. So the fatty acyl-CoA does not have its own. It uses that carnitine shuttle. This liver enzyme is only found in the mitochondria. It's going to be the HMG-CoA lyase. A couple slides on the, this is a good one to know, slide 86. It's about where everything happens that we've talked about in this unit. And slide 52 mentions these ketone body enzymes specifically. Which metabolite is not synthesized inside the mitochondria? Acetone is the weird one. So acetone is non-enzyme catalyzed. It just happens sometimes. The ketone bodies are synthesized, but for those, think about acetoacetate plus the hydroxybutyrate. Everything else, Krebs cycle, oxidative phosphorylation, Krebs cycle, all of those are definitely inside the mitochondria, plus the two ketone bodies would be as well, but not acetone. So number 17, which enzymatic step is not catalyzed by fatty acid synthase? That is hydration. So fatty acid synthase loads, goes through the condensation reduction, dehydration reduction, and then releases or repeats these steps. None of those steps are the hydration. Look for 65 for all of the fatty acid synthase enzymatic functions. Which molecule is the most immediate precursor for the synthesis of palmitate and cholesterol? So malonyl-CoA is for fatty acids but not cholesterol. HMG-CoA is for ketones or cholesterol, but not fatty acids and cholesterol. Don't care much about citrate here. Acetyl-CoA is the one that's for either palmitate or cholesterol. Which molecule is not a source of energy for brain cells? So anything with the eicosanoids or quinodinate don't have anything to do with energy production. Brain cells can use ketone bodies. You can use acetyl-CoA, anything produced in glycolysis. And then the Krebs cycle and oxidative phosphorylation. Which enzyme is not used to convert mitochondrial acetyl-CoA to palmitate? That's PEP carboxykinase. Saw that during gluconeogenesis. HCC is a big one in fatty acid synthesis, which is what this is talking about here. These are both in the citrate shuttle, and fatty acid synthase is the main player. So question about regulation. Which one is not regulated? Fatty acid synthase. All these other ones are discussed in 97 and 98, and if you're only on Unit 10 right now, I believe HMG-CoA reductase is only talked about in Unit 11, so don't let that one throw you off. Which metabolite is not part of the citrate shuttle? Malonyl-CoA is your metabolite used in fatty acid synthesis. Everything else is on the shuttle. I think this is the visual of it, and this is the steps written out, so it's good to know. Good to know both there. So metabolite is synthesized in all cells. Oops, hopefully you could have gotten this one in Unit 3. This is 2PG, anything from glycolysis. We're talking about synthesized here, so that's fatty acids and the ketone body cholesterol. Metabolite, which is in liver only. These two are liver only. And then lactate is mainly your red blood cells or muscle during vigorous exercise. Which reaction takes place only in the liver mitochondria? If you go through the reactions on this, A is the answer. So you're producing a ketone body. This is the only one where you're producing an actual ketone body. So that's how you know it's only taking place in the liver. If it's using a ketone body, you know it can't be the liver. If it's the reaction that makes hydroxybutyrate, that could be another one that only takes place in the liver. Fatty acid synthesis, only liver. This is mainly talking about ketone body sort of stuff. Let's see, this lipid process takes place in most cells. We just said fatty acid synthesis is liver specific, so are ketone bodies, so is cholesterol. So these are all pretty specialized functions, but fat burning can be done by most of the body. How many molecules of CO2 are generated by fatty acid synthase during the synthesis of one molecule of palmitate? So if you look at the reactions, I think this is the net reactions, you'll see that palmitate, there's 7 rounds per condensation round. This is happening in the condensation phase. That's when you produce one CO2. So that's where those extra carbons are going if you're doing the math on that. This metabolite is known to partially activate ACC. On the second to last slide, I think, it's citrate. Which enzyme is inhibited by aspirin? He brings this up in Biochem 1. It's the cyclooxygenase. How many molecules of NADPH are needed to synthesize one molecule of palmitate from cytosolic acetyl-CoA? So for that, you can either look at the net reactions here. The answer is 14. Condensation reduction, dehydration reduction. Each of these reductions both require an NADPH. And this is happening 7 times. So 7 times 2, that's where you're getting your answer there. If it was 14 molecule or 14 carbon molecule, you would do 14 minus 2 divided by 2. So that would be 6 rounds. So 6 rounds, each using 2 NADPHs, then the answer would be 12. If there's 18, then it would be 16 NADPH. You get the point. Which fatty acid process takes place in the ER? It's desaturation. There's another slide where he mentions this on, but this is a good review slide. So that's all of Unit 10. I'll do Unit 11 while I'm here. Let's see, which glycolytic metabolite is the most immediate precursor, the backbone of triacylglycerides and glycerol phospholipids. You'll see DHAP pop up on both slides 3 and 4. Which molecule is not needed for the synthesis of glycocolate? These are the biosalts. Taurine is the other one. So all of these are needed for glycocolate. Which vitamin is not derived from isopentenyl pyrophosphate, if I pronounced that right? Vitamin B12. All these other ones are lipid soluble. So I'll make sure to be familiar with this slide here. On the third slide, talks about which reaction is not essential for acidic glycerolipids. The answer to that is C. I would watch the other video for a better explanation of this stuff. Which protein is missing in liver cells? Phospholipase D is a plant-only enzyme. Which metabolite is not an intermediate for the synthesis of cholesterol? Malonyl-CoA, and that's your fatty acid synthesis molecule from Unit 10. All of these other ones are listed through the process on slides 25 and 26. This slide has to do with gangliosides, the degradation of gangliosides, not the synthesis. So make sure to know that. It's a pretty detailed question. compared to some other clinical ones he's asked before. So which vitamin is used in the synthesis of palmitate, cholesterol, glycocolate, and acetoacetate, which is one of the ketone bodies from Unit 10. The only one used for all of that is pantothenate. Remember, CoA has been the major player in a lot of these. We haven't been seeing our friend niacin as much lately. Biotin is carboxylase only. I don't think we've even mentioned folate yet. Riboflavin. Haven't seen much of that one lately either. Which signaling molecule cannot be synthesized from membrane lipids? Remember, membrane lipids is code word for cholesterol. It's on slide 34. The answer is vitamin A. Which molecule does not appear to regulate HMG-CoA reductase? That's citrate. Citrate's involved in the fatty acid regulation, all of this other stuff. You can see on the pathway on slide 28. So, study Unit 10 quite a bit. Unit 11 should come a little bit easier. And good luck on your test.